in-kind support for our Prairie Speaker Series is provided by Agriculture and Food Canada with Chantal, and providing the venue as well as the presentation today. And this project was also undertaken with the financial support of the Government of Canada through the Federal Department of the Environment. And I'm pleased to introduce Chantal. Chantal Mel has enjoyed working on the aeon symbiosis of croplands since her PhD research in the late 80s at McGill University. Her training background is soil science, which explains why she's careful not to walk on the soil, or as little as possible, whereas her plant science colleagues are careful not to walk on plants. So I always look down at plants, and I sometimes miss seeing bigger things. Now I have to look even closer and look at soil to pay more attention. She says that the soil is living and needs to be cared for. Producing lots of good crops and good return in a healthy environment is not an easy task, and we should make sure that the soil works for us. She's been the head of the Soil Microbiology Lab at Spark since 2003, so please join me in welcoming Chantal.
Thank you very much. Thank you everybody for coming to my talk. The microorganisms that live in the soil are very important. In fact, they are a determinant of the chemical equilibrium of the planet. And the way we manage the land has a large influence on soil microorganisms. I think there should be more interest into the field of land management and soil microorganisms, especially at this time in the time of the planet, because we have lots of environmental problems. Global change is a big one. In fact, the Arctic is melting, and it seems that nobody cares. So I found something very interesting lately that I would like to share with you, and it is the optimism bias. The optimism bias is a bias that makes people think that the future is going to be much better than the past or the present. And it's bias that makes people underestimate the likelihood of experiencing a negative event. For example, there was a study that was published in 2007 that showed that 70% of the respondents believe that families these days are less successful than in the time of their parents. But the same respondents were thinking that the future of their own family was going to be good. So it seems that we are optimistic for our family, about how we drive, about how we interact with people, and about how interesting we are. It seems that according to the scientist that does this research, about 90% of the people have the optimism bias. So we would like to try with you today to test if this is very true. And maybe we can just please raise your hand if you think that your driving skill is in the lowest 25% of the population. Yeah, no, please be honest. So nobody.
Not lowest.
Yeah, lowest 25%. So nobody. Now please raise your hand if you think that your driving skill is above average. So 11 out of 12. So 12 out of 16. So we can see just there that we cannot all be above average. So that's true, this thing of the optimism bias. But we should be careful about the optimism bias because, in fact, we can have car accidents, and smoking can kill us, and climate change will certainly give trouble to our children and grandchildren. Now, I have this image here that is a bit dark for this light room, bright room, but everybody knows these photos, and And this is the Earth at night. And this is very, very good to show how the human population has proliferated in the last 100 years. We are 7.3 billion people on Earth right now, and this is more than 3 times as much people we were when I was born. That's a lot. And when my granddaughter that is going to be born in February will be 25-year-old, there should be 9 billion people on the planet. So that's a lot of people. And my question here is, how optimistic should we be about our family or the future of our family? We are 7.3 billion people, and close to half the land on Earth was converted to cropland or to pasture land. And agriculture is the first cause of anthropogenic emission of greenhouse gas. So I think that we should think more about the way we use the land. So agriculture influences the climate, and it does that in different ways. One way is the production of nitrous oxide. Nitrous oxide is a greenhouse gas that has global warming potential 310 times more higher than the CO2. And the number here is from the US. So synthetic fertilizer will be responsible for 75% of the anthropogenic emission of nitrous oxide. And this is how it works. So we have ammonia in the soil, either from fertilization or from the decomposition of soil organic matter. And this ammonium in the soil is transformed into nitrate by soil microorganisms, soil bacteria, like urea nitrospora, which are organisms that are very active in the transformation of nitrite to nitrate. This nitrate in the microsite of the soil, where conditions are anaerobic, is transformed into nitrous oxide and dinitrogen. Dinitrogen is an innocuous gas, but nitrous oxide has a global warming capacity. And the pasture also produces greenhouse gas. It produces methane. Cattle grazing is responsible for about 25% of the emission of methane from anthropogenic source. And this is how it works. So we have carbon dioxide in the atmosphere that is taken up by plants through photosynthesis. So the plant grow in the pasture, and the cattle eat the grass. And cattle, they have a special microflora in their rumen that allow them to live on grass. So these microorganisms in the rumen of the cattle is able to decompose the fiber in the grass and make the grass assimilable by the cattle. But a byproduct of this digestion process that is done by bacteria, you can see here, is methane. So this is a source of greenhouse gas. But at the same time, these cattle have a very important ecological role. They replace the bison in the North American Great Plain, and their role is to maintain the soil organic carbon stock. And this is how it happens. So the cattle go and eat the tall vegetation, and by removing this tall vegetation, this encourages further plant growth. And at the same time, the cattle transform the vegetation they eat into fertilizing manure that further encourage plant growth. And when there's more plant growth, there's more carbon input into the soil. And research has shown that too little or too much grazing will reduce the soil organic carbon stock. And when soil carbon is reduced, it means that carbon dioxide is emitted from the soil, and carbon dioxide, as we know, is also a greenhouse gas. So depending on the management of the land, of the pasture, the soil will be a source or a sink of carbon dioxide. So the consequence of soil carbon loss, there are three main consequences. We have reduced soil productivity, because when we have less carbon, the soil physics is less good, water retention is less good, and plant productivity is less. By the way, this is a pasture that suffers from overgrazing, and this is what a healthy native prairie should look like. So in addition to reduced plant growth, we have, of course, carbon dioxide emission from the soil when the carbon stock of the soil is being depleted. And we also have a reduction in microbial activity. And this is quite interesting, because if you ask me, what's the consequence of reduced soil microbial activity? I'm sorry, but I'm not sure. I'm not sure. We know that some microorganisms are very important in mobilizing nutrients for plants, and many of them promote plant growth. They recycle the nutrients in dead organic matter. They will improve the porosity of the soil, improve water infiltration. They detoxify a lot of different compounds, agrochemicals, and other contaminants. But how this all is occurring is not always very clear, and not knowing, doesn't provide very good argument for soil conservation decision, especially in the face of the optimism bias. The problem with soil microbiology is that we did relatively little progress in this field for two main reasons. The first is probably the very huge diversity that exists in the soil. For example, in this handful of soil, for sure we have maybe more bacteria than people on Earth. We may have easily 50,000 species of bacteria in this soil. So the soil microbial diversity is very large, and this is a very complex system. And as you can see, to hold so many organisms in such a small volume, the microorganisms are very small, so we cannot see them. Most of them are unculturable, so the lack of means of study has been a big problem, preventing the progress of soil microbiology. However, since a few years, there are new methods that are quite powerful to study soil microorganisms, and this is what we use to kind of provide more knowledge, hopefully in support for conservation, soil conservation decision. This method, I will not spend too much time. It's based on we extract DNA from the soil, or any nucleic acid can extract from the soil, and with high throughput sequencing, looking at the sequence of the nucleic acid, comparing with data bank, we can draw a global picture of soil microbial communities. So that is what we use to try to address what are we losing when we lose a native prairie. So we went out and we compared the taxonomic and functional diversity in the soil microbiota under 4 different land use types. So of course a native prairie, crested wheatgrass pasture, cropland, and roadside. And I should mention here that roadside, is not the very side of the road, is the ditch, where we have, depend on where you are, but several meters of land that is not cultivated, can be harvested for hay, but it is a land that is undisturbed. And we went in the southwest of Saskatchewan, south of Swift Current. 5 locations were identified where the 4 different land use types were close to each other. And I'm going to show you from closer. I have a photograph here from the maize field site 2 to show you that the 4 land use types are here. So this is a native prairie, cropland, roadside, and crested wheat grass pasture. So we went out in the spring and we collected the soil samples at 3 depths, 0 to 15, 15 to 30, and 30 to 60 centimeters. And we analyzed the soil and look at the diversity of microorganisms, look at the diversity of soil bacteria, fungi, and mycorrhizal fungi. Here we are looking at the result that we got from the impact of land use on soil bacterial community. And if we focus on this panel here at the left of the screen, we are looking at the similarity or difference of the community of soil bacteria in the different land type used. So in yellow here, for example, we have the community that is selected under cropland. And the circle, blue and green here, represent the community selected under the native prairie in green and under crested wheat grass pasture. And because it is far apart, we know that the community under these different land use types are different. So all that to say that the most different community from the native prairie was that found under the cropland. This figure here shows that we found a trend for reduced diversity of bacteria under cropland. And here, I don't know if you can see very well, but it is community evenness. So the community seems to be less even in the cropland. And what that means is that the dominant bacteria, dominant species in the cropland are more dominant, and the subordinate species are even less abundant. At the bottom of the screen here, I put the groups where we found significant effect of land use. And we have the actinobacteria. Actinobacteria are some important bacteria. Many of them produce antibiotic, and several of them have plant growth promoting ability. So the abundance of the actinobacteria was reduced in cropland. The abundance of firmicutes was also reduced in cropland. The firmicutes are bacteria that are tolerant to desiccation. They include, among the well-known bacteria, the bacillus, which also often are beneficial to plants. The Gemeti monaletes and the nitrospirae are 2 groups of bacteria that were increased in abundance in the cropland. And this is quite interesting because the first group contains lots of bacteria that accumulate phosphorus. And the second group are the bacteria that I pointed out at the beginning that are very active in transforming ammonium into nitrate. So this indicates to us that probably soil fertilization has quite a larger impact on soil microorganisms than we may have thought in the past. So the proteobacteria is a very large group, very diverse. So it it was increased in the cropland. We don't know exactly what that means. And the last group, the vericomicrobia, we don't know much about this group. Perhaps the thing we know is that they are abundant in the prairie system. The abundance of these bacteria were influenced under roadside condition. Here we have a similar thing for the fungal community. The community of fungi were influenced by land use type. Again, the most different community of fungi was found in the cropland. We have here cropland, here native pasture. And again, we found a trend for reduced diversity here of the soil fungal community in the cropland. So at that time, it was richness. Richness is the number of different taxa that we find in a soil. So it's probably indicating that we are losing some taxa on the cropland. We have here the different groups that were significantly influenced by land use type. The first two groups are fungi that can grow under recalcitrant material, and we can see that under the roadside situation there was a shift in their abundance. The third group of fungi are fungi that we don't know very much what they're doing. But the fourth group, the Leotiomycetes, are fungi that interestingly interact with other living organisms. They can be biotroph, they can be pathogen, they can be endophyte, they can be mycorrhizal fungi, they can also be saprotroph. So the abundance of this group was increased under cropland. The abundance of the Hippocreales was also increased under cropland. The Hippocreales is well known for the Fusarium. Fusarium is a member of the Hippocreales, and as we know, it's a famous group of pathogen. But on the other hand, the Trichoderma are also Hippocreales, and the Trichoderma are antagonists, and they were used as biocontrol agents. The last two groups of fungi, the Soldariales and the Mortierellales, are saprotroph, of which we don't know much, but perhaps that the last group is abundant in the prairie. And we can see a shift also in the cropland. So overall, we can see that cropland, has a lot of influence on the structure of the fungal community. The mycorrhizal fungi were quite affected by land use type. And the mycorrhizal fungi, for those who don't know, are fungi that are important because they form a symbiosis with plants. So we think that they are important for plant growth. So that's why we have studied especially these mycorrhizal fungi. And we can see in this slide that also soil depth has an influence. So the white symbols here represent the community of mycorrhizal fungi under the different land use type in the surface layer of the soil. So we can see that the divergence of community structure is more marked in the topsoil here than in the deeper soil if we look at the darker symbols. Something that's interesting from this slide too is to see that the shift in community of mycorrhizal fungi in the cropland was related with the abundance of nutrients, and not only with the abundance of nitrogen and phosphorus, but also with the abundance of the metallic micronutrients, iron, manganese, and zinc. And this effect may be due to the fact that the use of nitrogen fertilizer acidifies the soil. As you can see here, the relationship was negative with pH. more the availability of the metallic micronutrients. If we look at the right-hand side of the screen, we can see that mycorrhizal fungi were also impacted by land use, and they undergo again a loss of diversity. And in this case, we can see that we had significant effect for richness and evenness. So it means that in the cropland, we're probably losing taxa of mycorrhizal fungi, and the taxa that are abundant are very abundant, and the ones that are subordinate are more rare than in other type of land use. Three groups were significantly affected by land use. The first group is the glomus, and we can see that they were less abundant, less predominant in the cropland, and this can be a good thing, in fact, because we found in our study that the glomus are not so good mutualists. At least we found that for wheat and for chickpea. The septoglomus were favored in the cropland, in the roadside, and the rhizophagus were decreased in the cropland. And this is a bit sad because our study also showed that rhizophagus are pretty good symbiont, at least as we have seen with Elijah, for wheat and for lentils. So this is about the data I have to show for the diversity. We also look at taxonomic diversity, the impact of land use on taxonomic diversity of soil microorganisms. But we also wanted to know what's the effect on function. So at the time of sampling, we took some undisturbed cores of soil that we can see here, and we planted flax in these cores. So the flax was growing in the greenhouse, all under the same condition, and we looked at the biomass production of flax in the different soils. And we can see in this graph here that the most productive soil were from the roadside, and the least productive soil were the cropland. There was no significant difference between the native prairie and the roadside. And maybe we can find interesting that the highest productivity is found in the soil of the roadside, especially if you have observed how roads are constructed. So the soil is just taken out, so the topsoil is taken out from the ditch, really. That's what the roadside is. But the ditch is also the place where the topsoil that is being eroded is going to end up, and this is where the fertilizer is also going to end up. So, as we can see, this may have an impact on the productivity of the soil. Another greenhouse experiment was conducted with the microbial community that was selected under the different type of land use. And this is what we see here. We have the cropland, crested wheat grass, native prairie, and roadside. So the microorganisms living in this soil were extracted and separated into mycorrhizal fungi in orange and other microflora. We could separate this because mycorrhizal fungi produce very big spores that could be teased out from the rest of the microflora. So we use this microorganism to inoculate different plants, wheat, crested wheat grass, and lentil that were grown in the greenhouse like this. And it was interesting to see that we grew best with the microbial community selected by crested wheat grass. Crested wheat grass grew best with the microbial community selected by the native prairie, and lentil grew worse with microorganisms selected in cropland and in crested wheat grass pasture. So that shows that different plants will respond differently to different microbial community. And this gives also some kind of explanation for the so-called rotation effect. Some crop will do better after some plant than after other, and this may be an effect of the selection of microorganisms by a crop, and then the effect of this microbial flora on the next. So this is kind of the end of my talk, and I would just like to summarize the problem again. So we have little understanding of how land use impact soil function, and in particular soil microbial communities. So this gives us little argument to support soil conservation decision. And this, with the optimism bias, makes that the soil and the environment is undergoing degradation. So I've shown that there is modification of the soil microbial diversity with intensification of land use, and this modification is concurrent with a change in soil productivity. I've shown that the microbial community are especially altered under cropland, and also we have found that fertilization has quite a larger effect than we thought on the shift of microbial community as we change land use. Also, I suppose you have understood that we still have a lot to discover on the effect of land use on soil microorganisms. And I would like to finish by presenting the sampling crew. So, and also Luc Benard, that is here with us today, who in fact did almost all this work all by himself. And Kerry Laforge, which is not here today. Her son is sick, but without Kerry, we would not have been able to find a sampling site, and this study would not have been possible. And here's Bill Houston, who actually initiated the development of this project. Heather was a student, a senior student working with us, and she was quite effective in directing the sampling crew. And this is me. And I would like to point out that when this photo was taken, the world population was just reaching 7 billion, and since then it has grown 0.3 billion. So I just have to thank you very much for your attention, and if you have any questions, be happy to answer, probably with the help of Luke and also of Brian right beside, that is quite more knowledgeable than me in terms of greenhouse gas. So, any question? Yes?
The roadside, that's quite a heterogeneous area, but you picked right at the bottom of the ditch then for your...
Yes,
okay.
yes.
So I have a little bit of optimism with that then, because obviously that, as you say, removed all the topsoil, and yet it recovered a lot of its function and microbial diversity over time.
But, you know, I don't know how old are these roadsides. Maybe somebody living here can tell me. But when I was sampling around the province, I've seen, because I was also thinking, the roadside is a very nice place. Sometimes it's very diverse. You have lots of plant diversity depending where you are. But I saw how they were redoing a ditch, and I was thinking, oh my God, so how can this be? But yes, that is true, that when you have erosion, this is where everything will end up. The soil, the topsoil is what is being eroded, and this is the best fraction of the soil. And also, you have, I must say, in the ditch, many ditch, and all the ditch that we sampled for this study was brown grass. And brown grass is a very productive plant, very productive. And when it is in the ditch, this is where the water goes. So you have lots of biomass production. And this probably has something to do with the soil being more productive there. Just a fertilizer carried with the runoff water.
So based on your preliminary results, do you think there'd be any benefit for cropland adjacent to native prairie then, from a symbiotic standpoint, like a production standpoint maybe?
Well, maybe the first thing I can think of is that the native prairie will be a place where diversity is. So if we're losing diversity, because now we don't know what's important, but maybe at some point we will find out that some things are important and we may have lost this diversity in the cropland. So we could always recover it from the prairie. And also, as I try to explain, the main cause of greenhouse gas emission from human activity, if I understand correctly, and Brian can help me with this one, but I think it is the change in land use, like deforestation and the conversion of a native prairie into cropland, because then you lose lots of the carbon that is accumulated in the soil. So that could be a big impact.
So, do you have information about, let's say, the geo-tillage or millennium tillage that is now more popular than the conventional, and if there is any difference in microbial diversity?
Well, we did not personally look exactly at global diversity. We look at mycorrhizal fungi. And the mycorrhizal fungi, they are biotrophic, meaning the plant lives. So if you have cropland, you have a crop just for some months, so it's not very good already. And if you destroy it, you break because fungi, we think microorganisms are small, that's true. But fungi, they grow as filaments. So in fact, they can be very large, they can be huge, they can be 1 hectare. Because it's a long pipe. And if you break it, you make it more fragile, less healthy. And then these pieces of fungi have to reestablish a symbiosis with the plant, so it will at the end make them be less abundant. However, it all depends on the depth of tillage, because these fungi will be more on the surface. And so if you till just like people do in the prairie here, just like 10 centimeters, this is very shallow. So you just destroy a little bit, but you have a big mass of fungi that remain. For the other organism, the major thing is that tillage will reduce plant biomass production in the prairie, because it will remove some of the water that is in the soil. The soil will dry, the soil surface will dry. So with less water, you have less productivity, because water is a limiting element in the prairie. So with time, we'll have reduced carbon input to the soil, reduction of soil carbon, and microorganisms. The biomass is correlated with the amount of soil organic carbon.
And mostly the minimum tillage is associated with roadsides, which have negative impact.
Yeah, that's again a very nice question for which I don't have an answer. But I do have a little bit of an answer. In fact, we looked at glyphosate, effect of glyphosate. And what we found was interesting is that chlorosmosate, to tell you the whole story, chlorosmosate was associated with glyphosate application. So when we put glyphosate in a soil experiment we did here, in the farm here, there was more Clovis morse. And Clovis morse, interestingly, is a mycorrhizal fungus that you see very abundant in cropland. If you want to see Clovis morse, you go in cropland. You will find Clovis morse will be probably the most abundant species you will find there. So maybe glyphosate will increase the Clovis morse, but it may be good for Clovis morse, but it may not be good for the other mycorrhizal fungi, because maybe the Clovis morse take the place of other fungi that could be better or worse for plant growth than Clovis morse. We know also that glyphosate has a mode of action that's quite interesting. People think it's not very toxic because it's not toxic for mammals, because we don't have the pathways that with which glyphosate interferes. But microorganisms have these pathways. But some microorganisms have a way to detoxify the glyphosate. So the ones that can detoxify glyphosate can even grow on glyphosate. But the ones that cannot detoxify glyphosate are negatively impacted. So glyphosate change community structure. It increase the proliferation of some and decrease the proliferation of others. The end result of this, we're not quite sure.